Hello everyone, welcome back. Today we are going with biological classification 25 question part 1. Let's do it. Question number 1 says 5 kingdom system of classification suggested by R.H. Whittaker is not based on, and the options are presence or absence of well-defined nucleus, mode of reproduction, mode of nutrition, and the last option is complexity of body organization. This question was asked in 2014 and the right answer is B. Mode of reproduction. R.H. Whittaker considered complexity of cell structure and structural body organization, mode of nutrition, ecological lifestyle, and phylogenetic relationships for the 5 kingdom system of classification. Now the next question is maximum nutrition diversity is found in the group fungi, Animalia, Monera, or Plantae, and the answer is Monera. Compared to many other groups, monerans, that is bacteria, show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs. They do not synthesize their own food but depend on other organisms or on dead organic matter for food. Now, next slide. Okay, here the question says in the 5 kingdom classification, Chlamydomonas and Chlorella have been included in, and the answer is the options are first Protista, Algae, Plantae, and Monera, and this was asked in mains 2012, and the answer for this question is A for Protista. In order to develop phylogenetic classification, R.H. Whittaker in 1969, an American taxonomist, divided all the organisms into 5 kingdoms. Whittaker has used 5 criteria for delimiting the different kingdoms: complexity of cell structure, prokaryotic and eukaryotic; complexity of body structure or structural organization, unicellular and multicellular; mode of nutrition, which is divergent in multicellular kingdoms; ecological lifestyle, like producers, Plantae, decomposers, fungi, and consumers, Animalia; phylogenetic relationship. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, Chlorella, earlier placed in algae within plants and both having cell walls, with Paramecium and Amoeba, which were earlier placed in the animal kingdom, which lacks cell wall. It has put together organisms which is earlier classifications were placed in different kingdoms. This happened because the criteria for classification has changed. Now, next question. In which kingdom would you classify the archaea and nitrogen fixing organisms if the 5 kingdom system of classification is used? And the options are Plantae, fungi, Protista, and Monera, and the right answer is D for Monera. Why? The kingdom Monera includes all prokaryotes. They are basically unicellular. The archaea and nitrogen fixing organisms are placed under Monera. All others, fungi, Plantae, Protist, and Animalia, are eukaryotic. Now, here next question says, in 5 kingdom system, the main basis of classification is a structure of nucleus, mode of nutrition, a structure of cell wall, or asexual reproduction. 2002. This question was asked in 2002, and the answer for this would be B. B as in mode of nutrition. This one. Okay, why? Whittaker system is based on the following 3 criteria: complexity of cell structure, complexity of body organization, and mode of nutrition. On the basis of these criteria, Whittaker divided organisms into 5 kingdoms. These 5 kingdoms are Monera, Protista, Algae, Fungi, and Animalia. In the 5 kingdom classification, all prokaryotes have been placed in kingdom Monera, all unicellular eukaryotes in kingdom Protista, fungi except slime molds and water molds in their separate kingdom, while kingdom Plantae and kingdom Animalia have been retained for multicellular, autotrophic and multicellular holozoic organisms respectively. Next question. In the 5 kingdom system of classification, which single kingdom out of the following can include blue-green algae, nitrogen-fixing bacteria and methanogenic archaebacteria? And the options are Plantae, Protista, Monera and fungi. This was asked in 1998 from this chapter, and the answer for this will be C, that is Monera. Okay, here it goes, Monera. And because R.H. Whittaker had proposed a 5 kingdom system of biological classification in 1969, it is based on complexity of cell structure, body organization and mode of nutrition. The kingdom Monera includes all prokaryotes. The blue-green algae, nitrogen-fixing bacteria and methanogenic archaebacteria are all unicellular prokaryotes, so they are included in the kingdom Monera. Okay. Next question. I'm sorry. BGA, blue-green algae, are included in which of the following groups? Bryophytes, prokaryotes, protista or fungi? And the answer will be prokaryotes. An important criterion for modern day classification is resemblance in morphology, anatomical and physiological traits, breeding habits, presence or absence of notochord. And the answer will be anatomical and physiological traits. Okay. Next question says, match the organisms in column 1 with habitats in column 2. Column 1 is halophiles and habitat is column 2 is column 1 is organism and column 2 is habitat. So here we have to match it. So for A, halophiles, it goes to salty area. This is the one. For thermoacidophiles, it goes to hot springs. Therm means hot, temperature, it is related with temperature. Number C, methanogens. Methanogens are in guts of ruminants. And number D, cyanobacteria is in aquatic environment. So A is with 4 and B is with 1. A4, B1. Okay, I think this is the correct answer. Perfect. Now, question number D. Which of the following are found in extreme saline conditions? Eubacteria, cyanobacteria, mycobacteria and archaebacteria. And the answer will be D, archaebacteria. Because halophiles, a type of archaebacteria, usually occur in extreme saline conditions like salt pans, salt beds and salt marshes. Okay. Now next question, 11th. Which among the following are the smallest living cells known without a definite cell wall? Pathogenic to plant as well as animals and can survive without oxygen. And the options are Pseudomonas, Mycoplasma, Nostoc, Bacillus. It was asked in NEET 2017 and the right answer is B, that is Mycoplasma. Mycoplasma are the smallest living cells known without a definite cell wall. They are pathogenic to both plants and animals and can survive without oxygen. Next question. Which of the following components provides a sticky character to the bacterial cell? Options are nuclear membrane, plasma membrane, glycocalyx and cell wall. And it was asked in NEET 2017. And the right answer for this is glycocalyx. Glycocalyx is the outermost mucilage layer of cell envelope which consists of non-cellulose polysaccharides with or without proteins. It gives a sticky character to the cell. Now, next question. DNA replication in bacteria occurs and the options are within nucleolus, prior to fission, just before transcription during S phase, and it was asked in NEET 2017, and the answer for this would be prior to fission, this one. Okay, DNA replicates in bacteria just before they divide by fission. Okay, the next question is methanogens belong to eubacteria, archaebacteria, dinoflagellates, or slime molds. This was asked in NEET 2016, the second module. And the right answer for this is archaebacteria. Methanogens belong to archaebacteria. They include methane-producing genera such as Methanobacillus and Methanothrix. Methanogens are obligate anaerobes found in oxygen-deficient environments such as marshes, swamps, sludge, formed during sewage treatment, and the digestive systems of ruminants. Mostly they obtain their energy by reducing carbon dioxide and oxidizing hydrogen with the production of methane. Next question. The primitive prokaryotes are responsible for the production of biogas from the dung of ruminant animals include methanogens, eubacteria, halophiles, thermoacidophiles. NEET 2016, and the right answer is methanogens. Question number 16. The archaebacteria differ from eubacteria in cell membrane structure, mode of nutrition, cell shape, mode of reproduction. This was asked in 2014, and the right answer is the cell membrane structure. The archaebacteria are the ancient bacteria that include extremophiles like methanogens, halophiles, and thermophiles. They represent some of the most ancient of life forms that persist today. They have both eubacterial and eukaryotic characters besides the features unique to them. Their mode of reproduction, nutrition, and cell shape and size resembles a typical eubacteria. Their cell walls are made of variety of polymers but do not contain peptidoglycan, unlike eubacteria. Lipids of their cytoplasmic membrane are either linked, unlike eubacteria, which contain glycerol ester lipids in their cell membrane. Got it? Okay. Now, next question is, which of the following are likely to be present in deep sea water? Okay. And the options are blue-green algae, saprophytic fungi, archaebacteria, and eubacteria. This was asked in NEET 2013, and the answer for this is archaebacteria. Archaebacteria belong to a group of prokaryotic organisms called Monera. These include the methanogens, which produce methane, the thermoacidophilic bacteria, which live in extremely hot and acidic environments such as hot springs, and the halophilic bacteria, which can only function at high salt concentrations and are abundant in the world's oceans. Next question is, pigment containing membranous extensions in some cyanobacteria are pneumatophores, chromatophores, heterocyst, basal bodies. And the answer for this is chromatophores, this one, B. Chromatophore is a pigmented lamellar or vesicular structure that can be isolated from disrupted photosynthetic bacteria or cyanobacteria. Their plasma membrane may be projected in folds into the cytoplasm, forming lamellae that have therefore double unit membrane structure. The pigments and most of the enzymes required for the light-induced electron transport and phosphorylation process of photosynthesis are located in the plasma membrane and lamellae. Now, next question. I think just before one. Okay. Okay. Here it goes. Now, the next question is, the cyanobacteria are also referred to as The cyanobacteria are also referred to as protist, golden algae, slime molds, or blue-green algae. The answer for this is cyanobacteria is also referred to as is blue-green algae. Blue-green algae. Okay, cyanobacteria consist of two groups of photosynthetic eubacteria, the blue-green bacteria, formerly known as blue-green algae or cyanophyta, which comprise the vast majority of members, and the grass-green bacteria or chloroxybacteria. Next question is the most abundant prokaryotes helpful to humans in making curd from milk and in production of antibiotics are the ones categorized as. This question was asked in 2012, and the options are cyanobacteria, archaebacterial, chemosynthetic autotrophs, and heterotrophic bacteria. And the answer for this is heterotrophic bacteria. This one. Okay, and the maximum number of antibiotics are produced by mycelial bacteria known as actinomycetes, and most of the actinomycetes are saprotrophic, that is heterotrophic. Lactic acid bacteria that are used in preparation of curd are also heterotrophic ones. Okay, next question. In eubacteria, a cellular component that resembles eukaryotic cell is plasma membrane, nucleus, ribosomes, or cell wall. This question was asked in 2011, and the right answer is plasma membrane. Plasma membrane of eubacteria resembles plasma membrane of eukaryotic cells. Both nucleus, ribosomes, and cell wall are little different in eukaryotic cell in their structure and organization from eubacterial cell. Next question. Some hyperthermophilic organisms that grow in highly acidic pH 2 habitats belong to the two groups: eubacteria and archaea, cyanobacteria and diatoms, protists and mosses, liverworts and yeast. What is the answer? The correct answer is A, eubacteria and archaea. There are two major groups of modern and archaebacteria, ancient bacteria and eubacteria, that is true bacteria. Eubacteria is of further two types, bacteria and cyanobacteria. Thermoacidophiles are a type of archaebacteria which live in extremely acidic environment pH 2 that have extremely high temperatures up to 110 degrees Celsius. They are found in hot sulfur springs. Some of the eubacteria are also famous for living under the most hostile environment like salt pans, petroleum pans, spilled oil, hot springs, sulfur springs, snow, etc. Okay, now next question 23rd says select the correct combination of the statement 1 to 4 regarding the characteristic of certain organisms. Okay. Okay, select the correct combination of the statements. Okay, there are 4 statements and whatever is correct should be entered. Okay, the statements at number 1 says methanogens are archaebacteria which produce methane in marshy areas. Next statement says nostoc is a filamentous blue-green algae which fixes atmospheric nitrogen. Statement number 3 says chemosynthetic autotrophic bacteria synthesize cellulose from glucose. And the statement number 4 says mycoplasma lack a cell wall and can survive without oxygen. And the correct statement, whether it is 2 and 3 are correct, or 2, 3, 4 are correct, 1, 2 and 3 are correct, 1, 2 and 4 are correct. For this, it is 1, 2 and 4 are correct. 1 is correct, 2 is correct, and 4 is correct. This one is not, the third one is not correct. Okay, so 1, 2 and 4 is in D, 1, 2 and 4. So here the answer would be D, and this question was asked in May 2010. Next, second last question, 24th question. Bacterial leaf blight of rice is caused by a species: Alternaria, Erwinia, Xanthomonas and Pseudomonas. This was asked in 2008, and the right answer is C, Xanthomonas. Bacterial leaf blight of rice is caused by Xanthomonas oryzae, a bacterium which is gram-negative, aerobic, capsulated and motile with a single polar flagellum. Now, the last question of this video is Thermococcus, Methanococcus and Methanobacterium exemplify. And the options are bacteria whose DNA is relaxed or positively supercoiled, but which have cytoskeleton as well as mitochondria. Second is bacteria that contain cytoskeleton and ribosomes. Third option, archaebacteria that contain protein, homologous or eukaryotic core histones. And the last option is archaebacteria that lack any histones resembling those found in eukaryotes, but whose DNA is negatively supercoiled. And the answer for this is C. That is, Thermococcus, Methanococcus and Methanobacterium exemplify archaebacteria that contain protein homologous to eukaryotic core histones. Okay. Are examples of archaebacteria which are characteristic by a unique cell wall that lack peptidoglycan and consist of polysaccharides and protein, and closely resemble the eukaryotic cell in the mechanism of protein synthesis, a structural protein and RNA complements of the ribosomes. So here it goes the end of the video. 35 questions are done. Next 25 will be in the second part of this video. Any comments? Please write down in the comment section. Thank you very much.